everyone. I'm Brooke. I did my summer research on the microhabitat selection of bird voice tree frogs down in southern Illinois. We are concerned about these tree frogs because they're a state-threatened species. This is because they are habitat specialists for swamp habitats, which unfortunately are disappearing due to human activities, such as wetland impoundment that is altering the hydrological conditions of these swamps and making them disappear. And so the goal for my project was to learn about their microhabitat preferences in order to aid in future conservation efforts and hopefully implement those characteristics into environments that we wish to conserve the species in. And so specifically, I was looking at the microhabitat preferences concerned with the tree species and tree size, and if that preference changed across site qualities, which I'll explain later. But this is the bird voice tree frog. It's called, sound like this, if you can hear it. Okay, you can see where it gets its name. It kind of sounds like a bird. Hyla evivoca, and then we also collected data for the gray tree frog complex, Hyla versicolor and Hyla chrysosceles, and the green tree frog, Hyla cinerea, but those are very common species, so they were just collected opportunistically. I had a couple hypotheses going into this study. So the first one was that I would find more frogs on bald cypress and tupelo trees. This is just because these trees are associated with really high quality swamps, so I figured that these were the more high quality trees and we would find more frogs on them. And then I also hypothesized we would find more frogs on pristine sites rather than degraded sites, so higher quality versus lower quality sites. These were all of our sites during the study. We had 10 of them. The ones on the western side of the map near the Cache River are part of the Cache River system. And then the group on the eastern side of the map is part of the Eastern Shawnee system. The sites ranged by quality, so those are notated by the points. The diamonds are the pristine sites, the circles are the partially degraded sites, and the squares are the degraded sites, so the worst quality ones. This is an example of a pristine site. You can see that the canopy is very closed and dense. It's seasonally fledged, which means that there are seasons of complete dryness, which allows trees like the bald cypress and tupelo to establish themselves and germinate and grow bigger. This is an example of a partially degraded site. So the canopy is a little more open than the pristine site. It's semi-permanent flooding, which means the water recedes, but it never fully dries. Which allows for some bald cypress and tupelo to establish, but not as much as the pristine sites. And then lastly, we have an example of a degraded site, which you can see the canopy is very open. It's usually permanently flooded all year round, which does not allow a lot of those bald cypress and tupelo trees to establish themselves and grow really big like we wish. At each of these sites, we had 50 of these PVC pipe traps, which allowed the frogs to enter and exit them as they pleased. They weren't actually trapped in there. And each site had 2 transects or lines of traps that had the traps placed about 3 meters apart on the next random tree to try to avoid selection bias. So if you're doing the math, 10 sites times 50 traps equals 500 traps total for the study. So the first thing I did at these sites was identify all of the trees that the traps were on, and I mostly did this using the leaves and the bark of the tree. And then I took some measurements. So the first thing I measured was the DBH, which is what you see me doing in that first photo down there in my super cute outfit with that little tape measure. It's called DBH tape, and it tells me how thick the trees are, which usually correlates to their age. And then in the second picture, I'm using an electric hypsometer, which tells me how tall the trees are. We then checked traps. We checked between 50 and 250 traps a day. It just depended on how many sites we got to. We then administered a pit tag into all novel frog captures. The reason we did this was because I was only interested in taking data for novel frog captures, so we wanted to know if we got a recapture or not, and we figured out if we had a recapture using that little green scanner there to see if they had a pit tag. And then lastly, I analyzed data using our statistical software as well. These are all of the trees that I identified over all of our sites. These are USDA official codes for the trees, so that's what you're seeing there. But the most important ones are boxed in red for you. So we've got a lot of red maples, green ash, tupelo, and bald cypress trees. But there's everything if you're curious. So I used 2 data sets to analyze my results. The first data set is the data that I took this summer. So that's what you're seeing here. And this graph is showing the DBH, remember how thick those trees are, split up by site quality. So we've got the degraded sites on the left, which are the lowest quality sites, and then the partially degraded in the middle, and the pristine or the high quality sites on the right. And although there was no significant difference between these DBHs, you can definitely see kind of a pattern of a shift here. So in our degraded sites, we have kind of a right-handed skewed graph. So a lot of trees are between like 10 and 20 centimeters, and in the partially degraded, we see more of like a bell-shaped curve, where a lot of the trees are between like 15 and 25, and then we kind of see a pattern in the pristine sites of a left-handed skew, where a lot of the trees are between 20 and 30. These are all of the tree species that I found, again split up by site quality for my 2021 data. The ones that are most important are these top 4 right here in this box. And those are the tupelo, bald cypress, red maple, and green ash, if you need a reminder. A pattern to notice here is that we're seeing a microhabitat shift across site quality. So the most frequent trees in the degraded sites are that red maple and green ash, but you can see as we move to the pristine sites, the higher quality sites, we're seeing more frequent tupelo and bald cypress trees. So these are the frog captures for my 2021 data by tree species split up by site quality. So again, the top 4 most frequent trees that we saw in the last graph are also the top 4 most frequent that we're catching the frogs on. And again, we're kind of seeing that shift where we're finding more frogs on the red maple and green ash. And then as we shift to higher quality sites, we're seeing more frogs on bald cypress and tupelo. Something to notice here is that we had a very low sample size. We only caught 1 gray tree frog and 1 green tree frog, so I should have said this, but the blue is the bird voiced, the green is the green tree frog, the gray is the green tree frog. So we only caught 1 gray and green. The sample size was very small. This was probably due to the fact that we were collecting in the summer, which is their breeding season, and so instead of going into our traps, they were out in the swamps breeding. And a pattern that you should notice here is that we actually caught the least amount of frogs in the pristine sites, which is not what we expected. So then, because we had such a small sample size, I decided to use some historical data to do the rest of my analysis and it was collected from 2017 through 2019 over 3 seasons this time, so spring, summer, and fall data instead of just summer like mine was. And I made the same graph here. So these are the DBHs, or the size of the trees, split up by site quality. Again, no significant difference between site quality, but we're still seeing that skewness that we were seeing in my data, where we have smaller trees, or it seems like we have smaller trees in the degraded sites, and then as we get to the pristine, we're having, on average, bigger trees. These are, again, my frequencies of trees by site quality for the historical data. The same top 4 that we've been seeing, tupelo, bald cypress, red maple, green ash, and we're again seeing that microhabitat niche shift as we go across site quality from degraded to pristine. So more bald cypress and tupelo in the pristine sites. And then I just zoomed in on that for you so you can really see that shift just between those 4 trees. And then these are the frog capture data for the historical data set. Again, same top 4 trees that we're seeing. You can really see that shift for the red maple and green ash and the degraded sites catching more frogs, and then as we shift, we're catching more frogs on bald cypress and tupelo. And but again, we're seeing the least number of frogs caught on the pristine sites, which is the opposite of what we expected to happen. And then I just zoomed in on those top 4 for you so you could see that. In fact, the difference between the frogs captured on the degraded and pristine sites was so big that it was actually significant. So we ran an ANOVA test to figure out if there was significance. We found out there was, so then we ran this Tukey test to find exactly where that significance was, and it was between the pristine and degraded sites. Since those error bars don't overlap the 0 line, that's what that means there. So, to conclude, there was a significant difference between degraded and pristine sites. There's a few reasons why we think this happened, or could have happened. Number one, it could just be because pristine sites, higher quality sites, have more natural refugia for the frogs to hide in, and so they weren't going in our artificial refugia, whereas in the degraded sites, that might be the only refugia available. Two, it could be because the tree frogs aren't as affected by the swamp quality as we previously thought, and maybe they're more affected by things such as like surrounding terrestrial forest quality, which is something that we didn't collect data on, so in the future that may be something we look at. And three, if you remember the frequency data for the split up by site quality, there was a more diverse tree species in the partially degraded and degraded sites versus the pristine sites. So it could just be because there's more niches available, maybe more bug species available because of that. So with more food, we might be finding more frogs in those degraded sites. We had some limitations in this study. The big one was time, or the big two were time and sample size. So we definitely need to continue this study over multiple seasons to come to any real conclusions. There was also unequal site categories. So we had 6 partially degraded sites and we only had 2 pristine and 2 degraded. So I think if we got more pristine and degraded sites, that could eliminate any confounding variables with that. And we also had subjective categories when categorizing site quality anyway, so we might want to develop something that's more objective to categorize those. We also found that higher quality sites had more bald cypress and tupelo trees, which is what we expected. And in the future, we hope these results will help in future conservation efforts for the bird voice tree frog as the study continues through 2023. So just to revisit my hypotheses, I hypothesized that bald cypress and tupelo trees would have more frog captures, which it did, but also it was only really for the pristine sites, and those were the most frequent trees at those sites. So I don't think we can draw any conclusions, especially with the small sample size and time constraint that we had. And my other hypothesis was that we would find more frogs at pristine sites, which was not supported. We actually found the opposite. So I'd like to acknowledge all of these organizations for their funding and support, and I'd like to give special thanks to Catherine Nguyen, Jessica Molman, Dr. John Crawford, and Dr. Lyle Guyon for helping me through this summer. So I'll take questions now. Yeah, let me see.
Were you exposed to the historical data set before making predictions for your study?
No, I was not. So we were trying to see how many frogs we would capture first before we decided to use the historical data. And then when we didn't catch that many, we decided we should probably use it.
Yeah, you mentioned surrounding forest quality, like something that could be measured maybe in the future. I think your counterpart mentioned this too. Do you know that these frogs are using uplands after the breeding season, or are they just hanging out in the swamps all?
Yeah, so I think that whenever the water recedes and dries, they do use more of the forest because I'm assuming that's where a lot of their food is and stuff during that time. So, yeah.
Did my dendrology quizlet help you at all?
Yes, it did. I appreciate that. Thank you.
So, in each of these areas, degraded and partially pristine, it's the same number of traps you're putting. So, one thing wasn't clear to me, but from the pictures, it appears like there's fewer trees. The more degraded it is, there's fewer trees. Is that right?
In the more degraded, there are fewer trees. Yes.
So, kind of, if you thought about like, if you think of like traps per tree,
Mm-hmm.
that would kind of be different in each area, right? So essentially, there's more traps,
Mm-hmm.
fewer trees, more traps. I just wonder if that has any impact too on why you're seeing.
Yeah, that's a good question. I'm not sure. That might be a thing that we could try in the future, especially since we had those unequal site qualities as well. And maybe instead of finding more sites, we could just alter the number of traps that we have at each site or something like that. But they were, the degraded sites were able to still have trees 3 meters apart. So that was kind of
Oh, so
controlled
they're
for.
at equal distance?
Yes.
Oh, okay.
Yeah.
Nice. And then, I mean, do these frogs move around a lot from tree to tree, or do they?
I'm not really sure. I think that the males definitely do some more moving than the females, especially at this time of year. So we caught a lot more male frogs than females. But yeah, I'm not sure about that completely. All good? I'm going to pass it off to Josh.